All right, I'm audible at the back. Thanks, Tanweer, for that nice introduction. I would begin with thanking Umesh first and foremost because he was the one who almost 1.5, 2 years ago brought us all together, told us, let's do this. We had no idea, but I'm so glad I became part of it, we became part of it. It has been a wonderful learning experience. This is the first conference that I have been part of organizing, and it's been a lovely learning curve. So thanks, Umesh. All right, so I'm at the Department of Biochemistry at Indian Institute of Science. This is the administrative building of this institute. And what I'm going to share with all of you is a single unpublished story, right? And I'm going to tell you the gist of the story before I go into trying to convince you why we think what we think. There's an arginine methyltransferase by the name of HMT1 in yeast, and its human homologue is PRMT1. What we are observing based on the results is that this particular enzyme is involved in modulating the toxicity of alpha-synuclein, right? And we think this is a non-canonical role of this enzyme because it seems to be independent of its methyltransferase activity. This is the gist of what I'm going to tell you. So if you don't pay attention anymore, it's fine. This is what I'm going to try and convince you. All right, so this is our lovely campus. If you all liked ICTS, you all would love IISc. ICTS is kind of on the outskirts of Bangalore, but our campus is bang in the middle of the city, almost like an oasis. So for those of you who haven't managed to visit this campus and this visit, I hope you do try and manage to come in near future. All right, before I jump into telling you the details of that non-canonical role of the arginine methyltransferase, I'll take a few minutes trying to tell you what is it that we are excited about in general. What is it that we have been trying to do for the past 12 years since we set up the lab? So the overarching question in the laboratory is, how do low complexity sequences contribute to mRNA fate? And some of you might be thinking about two questions. What are low complexity sequences and what do I mean by mRNA fate? So let me take you through that. All right, so this is a sequence of a protein. It doesn't matter what protein this is, but I hope you will agree with me that there are lots of repeats in this sequence, right? And what that means is that all 20, 21 amino acids are not more or less equally represented, which is what happens in most globular folded proteins. You see overrepresentation of certain amino acids and therefore underrepresentation of other amino acids, right? So such protein sequences are considered to be those with low complexity, right? They do not represent the entire complexity of all amino acids in a given sequence. So these are the kind of proteins that we are interested in the lab, those that have repeats of amino acids of a specific kind. This is another evidence with a different kind of a repeat. Okay, so why are we interested in low complexity sequences? Well, often low complexity sequences, because of the less information that they have within them encoded, they do not tend to fold, right? So they remain in an unfolded form. They're also known as disordered regions in a protein. And as people have started looking for these kind of sequences and proteins and their function, it's becoming increasingly clear that they are way more prevalent than what it was thought to be, right? So in this particular review, it has been argued that almost 50 to 60% of the proteins have a certain constituent within them which is intrinsically disordered. And intrinsically disordered regions of a protein often have low complexity sequences. 5% of them are almost entirely disordered. And this goes against what we have read in the textbooks, right? Because structure is important for a protein function, whereas now we are learning that many proteins which do have structure, but they also have a certain portion within them that does not seem to have a structure, right? And this happens because these low complexity sequences, or the intrinsically disordered part, allows a certain level of promiscuity that allows these proteins to interact with a variety of different ligands, right? It could be proteins, RNA, DNA, small molecules, whatever it is. And therefore, it increases the gamut of interactions that these proteins can participate in. So it's almost like a Swiss knife, right? You will have one protein which based on its low complexity sequence, which leads to the disordered region, can interact with a variety of different things. So this is one of the motivating factors for looking at these sequences. All right, what is mRNA life cycle? All right, so more often than not, mRNA comes to the cytoplasm and gets translated. That doesn't happen all the time. That doesn't happen to all the mRNAs under all the conditions. Often, these mRNAs are stored in a form where they are not getting translated. So this is referred to as translational repression. And there are a variety of mechanisms that have been described for this to occur. Many a times, the repressed mRNAs are stored in these structures that we have heard about in these meetings. In this particular meeting, known as RNP condensates, some of the most well-characterized forms of these condensates are P-bodies and stress granules. So they could be going back and forth between these structures. And of course, these condensates can assemble and disassemble, and many a times these mRNAs can move out of it upon disassembly, and they can either return back to translation or they can get degraded, right? So many mRNAs can go through this kind of a life cycle where they are either getting translated, remaining stored, or getting degraded depending upon the kind of proteins that they associate with. And the most important class of proteins that determine this are RNA binding proteins. Okay, so we focus on RNA binding proteins with low complexity sequences in our lab. And for vast majority of the past 12 years, we have used yeast as a model system. We still continue to use yeast as a model system and would love to do that. But now we are also using human cells as our model system. All right, so these are different proteins. Names are not very important, but what is important is that all of them have this particular low complexity sequence, which is one of our favorite kind of low complexity sequence, which has arginine and glycine repeats. It's known as RGG motif. They also have other RNA binding domains, which can fold very well, right? For example, this protein has RRM, this is LSM, so on and so forth. So often you have a protein that has traditional three-dimensional folded domains, but they also have these kind of sequences which are low complexity sequences. I'm giving you just a glimpse of the low complexity RGG motif in each of these proteins. In green is arginine and glycine residues which have been highlighted. So you can see that these regions, which are actually very small part of this protein, anywhere from between 40 to 60 amino acids, and most of these proteins are 300 amino acids or more, right? So it's a very small portion. And you can appreciate the kind of low complexity or overrepresentation of arginine and glycine residues that's present in these proteins. So these are some of the proteins that we focus on in our lab. All right, so these are RGG motif containing proteins. But these same motifs could be methylated at the arginine residue. This is known, right? People have figured out that RGG boxes could be sites of a PTM known as arginine methylation. And upon undergoing arginine methylation, it regulates interaction of this motif with a variety of different ligands. It could be proteins, nucleic acids. So we have studied these proteins over the course of past 12 years, and we have looked at their role in translation. We have understood the functioning and mechanism of different RGG motif containing proteins that affect translation, predominantly translation repressors. So these are the proteins that prevent the translation of mRNAs, and they often play an important role in response to stress because translation repression is a conserved stress response. We have also looked at their role in RNP condensates in general, P-bodies and stress granules. And one of the important findings that we came across was that these low complexity sequences could also act as disassembly factor for P-bodies. This was unexpected because often these repeats are implicated in the assembly of these structures. We found an example where it is important for the disassembly. And we have also looked at the implication of these proteins in mRNA stability. I'm not going into the details of that. All right, so many of these things that we have looked at has explored the regulation of these proteins by arginine methylation, right? Now, what is arginine methylation? So you could see that the arginine in a given sequence, specifically in these RGG boxes, can undergo a single addition of a single methyl group, which is known as monomethyl arginine, or it could undergo addition of two different methyl groups. It could be symmetric dimethylation as shown here, or it could be asymmetric dimethylation. And HMT1 in yeast and its human homologue PRMT1, they catalyze mono and asymmetric. I'm sorry, this is a typo. It's not demethylation, it's dimethylation. Demethylation is removal of the methyl group. By the way, that's not known. So far, there is no protein known that removes the methyl group. We are working on that aspect, but as of now, there is no proof that it is reversible. Anyways, so this particular protein does this, and it does this, right? Okay. It turns out that RNA binding proteins are the largest class of arginine methylated proteins, right? RGG motif containing proteins are the second most abundant class of RNA binding proteins, and since these can get arginine methylated, it's not surprising that the largest class of arginine methylated proteins are RNA binding proteins. Okay, so arginine methylation on its own, right? So arginine methylation wouldn't just happen on RNA binding proteins, it can happen on a variety of other proteins as well. Has been implicated in both health and disease. There are PRMT inhibitors which are being under trials for different kinds of cancer. There is arginine methylation of different proteins implicated in neurodegeneration, for example, FUS, which seems to affect their phase separation and/or toxicity. It's been implicated in metabolic diseases and in context of aging. So as such, this PTM is also a very important modification, right? Over and above the fact that largest group of arginine methylated proteins are actually RNA binding proteins. Okay, so we were interested in understanding the regulation of this enzyme simply because a lot of our RNA binding proteins of interest were known to undergo methylation, right? Now, why did we think there is potential regulation or interesting regulation that could be underlying here? So that's because of this reason. So HMT1 is a predominantly nuclear protein. So if you took a strain that's tagged genomically with HMT1 and looked under the microscope by live cell imaging, you would find most of the protein in the nucleus. There will be some protein in the cytoplasm also, but it's concentrated in the nucleus. Now, there are many substrates of HMT1 and PRMT1 which are in the nucleus, but most of our proteins of interest, which are RNA binding proteins affecting mRNA fate, they are present in the cytoplasm. So that made us wonder that, is it possible that this kind of a localization changes under certain conditions? And that change in turn could affect the function of our proteins of interest, thereby affecting mRNA fate. So what we are looking for is something upstream of HMT1 that affects these proteins and their functions in the context of determining mRNA fate. So this work has been spearheaded by a very hardworking and talented graduate student, Sakshi. She's in the audience. Many of you probably have already met her. She started with the hypothesis that change in localization could modulate HMT1 function. So we started with yeast, of course. Okay, so to test this hypothesis, first thing first, does it undergo a change in localization, right? I mean, we didn't know that. So what she did was she expressed HMT1 from a plasmid on a wild type yeast, exposes to different kinds of stresses, let those cells undergo recovery, and looked at both the stressed cells and the recovered cells under the microscope. Okay, so this is how unstressed cells look like. You can see it's predominantly nuclear, but there is also some cytoplasmic signal. Under heat stress, there is pretty much no difference in the localization of the protein. She looked at hydroxyurea, again, pretty much nuclear localization along with the cytoplasmic localization, which was visible under unstressed. Same thing with nitrogen starvation, amino acid starvation, and also glucose deprivation. All of these stresses, by the way, they lead to global translation repression, and thereby you would expect that they would affect many of those RNA binding proteins of our interest because many of them are translation repressors. However, it turns out that the protein really doesn't undergo much change in localization. When Sakshi looked at oxidative stress, she observed that when she treats the cells with hydrogen peroxide, they delocalize from the nucleus and they are more localized to the cytoplasm, and they are present in the cytoplasmic puncta. The change was reversible. You take away the stress and the protein relocalizes to the nucleus, right? Suggesting that these are not endpoints, but perhaps there is a dynamic regulation that's involved. Another oxidative stress, to confirm that this was not just one weird stress, she looked at sodium azide, 0.5%. And again, similar thing, there is delocalization from the nucleus. Here, you pretty much don't see any nuclear localization, cytoplasmic localization in form of puncta. Again, it's reversible. You take away the stress, protein returns back to the way it was in unstressed condition. So this provided us very good tools, right? That the kind of regulation we were looking for perhaps is happening in response to specifically oxidative stress, right? Global translational repression occurs in almost all these conditions, but seems like HMT1 is responding to this specific stress. Of course, sodium azide and hydrogen peroxide also have many things different amongst them when you look at the signaling and things like that, but overall, these are oxidative stress. So this is the quantitation of that, the number of granules per cell. What I want to highlight here is that Sakshi wanted to make sure that this is not something because of just increase in protein levels of these cells under these conditions. So H2O2 is this. I would like you to compare it with unstressed. There is a slight increase in HMT1 GFP levels upon this stress, but in sodium azide, there is actually a decrease in the total protein levels as compared to unstressed. More importantly, all of the stresses actually lead to increase in the HMT1 levels, right? So despite that increase, HMT1 does not seem to undergo localization. So we don't think this is simply a function of increased protein levels causing localization of this protein to cytoplasmic puncta. Right, so HMT1 delocalizes from the nucleus, localizes to cytoplasmic granules under oxidative stress. That was plasmid borne. Sakshi wanted to make sure that this happens with endogenous HMT1, which is not being overexpressed. So that is what she did here. She looked at endogenously tagged HMT1 GFP in response to sodium azide 30 minutes, in response to H2O2. Again, they localized to cytoplasmic puncta, and it seems to be reversible. You take away the stress and protein returns back to nucleus. All right, now we were excited. Why? Because a lot of our proteins go to puncta in the cytoplasm, right? Condensates, P-bodies, stress granules. So you were like, wow, all right. This is the site where probably the enzyme and the substrates meet, right? It's a good way of regulating, ensuring that methylation occurs only under specific condition. HMT1 comes out of the cytoplasm, meets at this rendezvous site, methylates, and then goes back, right? So we asked the question that do these co-localize with P-bodies, which are one kind of RNP condensates, and we looked at EDC3. So again, the same sequence of events. What we observe is that there is pretty much no co-localization between the HMT1 puncta and the EDC3 foci that come up, suggesting that these are not really P-bodies. I'm not showing you here, but Sakshi observed similar observations in context with stress granule marker, indicating that these HMT1 puncta, by and large, do not co-localize with either P-bodies or stress granules. All right. Now, we were like, all right, they may not be classical P-bodies and stress granules, but if they are sites of methylation of our proteins, perhaps our proteins would co-localize with the HMT1 puncta, because we know that our proteins of interest also go to cytoplasmic puncta. Okay, that's not me. Okay, all right. So we looked at two different substrates of HMT1. I'm sorry? Somebody's there. Okay, we looked at two different substrates of HMT1, which we have characterized in our lab that they do get methylated by HMT1. They undergo monomethylation and dimethylation, SBP1 and SCD6. And what we again observed was that, again, green is HMT1 strain. We are expressing these proteins on a plasmid, and we find that neither SBP1 nor SCD6 puncta seem to co-localize with these HMT1 puncta that come up in response to stress, right? So that was a bit of a disappointment, because that hypothesis goes out of the window, that perhaps these punctas are unlikely to be sites of methylation, because we pretty much do not see any co-localization with either condensates in general or with two specific substrates that we have looked at. All right. So then one of the other things that Sakshi was very much excited about looking at after seeing these results was that, does this depend on the catalytic activity of HMT1, right? So is this localization change a function of its catalytic activity? So she created this particular mutant, which is in the SAM binding domain of HMT1, that's this purple region. People have characterized this mutation. We didn't make it for the first time, that G68R completely abrogates catalytic activity of HMT1 because it cannot bind SAM. SAM is the methyl donor for a methyltransferase. So she created this mutant, looked at its localization, again by the same scheme, and what she finds is that the catalytically dead mutant pretty much localizes to puncta in a manner that's comparable to wild type. If anything, it's actually slightly better than wild type in terms of localizing to the puncta, right? So this means that whether the enzyme is active or not, it likes to come to the cytoplasm and localizes to these puncta. This is the proof that the protein is actually completely inactive. What you're looking at here is actually not a molecular weight marker. There are 5 major bands that come up when you stain a yeast lysate with a monomethyl arginine specific antibody. And you're looking at 3 of these here, and I think the other 2 have cut out. But the blot is completely blank when you express, you take HMT1 deletion strain and put HMT1 G68R in that. This is one of the other mutants that she had made, and I'm not going into that, but just to tell you that it pretty much has no catalytic activity. However, it does localize to these structures. Okay, so now we got a little desperate, doesn't co-localize with condensates, doesn't localize with substrates, seems to be independent of its catalytic activity. So Sakshi came up with the idea that she will force localization of this protein to different parts of the cell, and then ask the question that if we did that forced localization, would the protein still come back to the cytoplasm? Is this coming to the cytoplasm and localizing to granules really important for this protein? So she made these 3 different kinds of constructs in one. So even though this protein localizes to the nucleus, it doesn't have any apparent nuclear localization sequence. So we still don't quite understand how this protein goes to nucleus. We do know that a certain portion still stays in the cytoplasm. So Sakshi's idea was that if I append a proper NLS, then it will be forced to go completely to the nucleus. And in that case, what happens? Does it still respond to stress the way the wild type does? The other was the opposite thing. Let's append a nuclear export signal so that it stays in the cytoplasm all the time, which would make sure that perhaps it would go more to these puncta because now the resident time in the cytoplasm increases. And the other one was where she appended a specific sequence that tethers the proteins to the cortex of the yeast cells. So the idea was that even though it's in the cytoplasm, you're taking it somewhere and you're almost tying it to the cortex and then asking what happens. So she made these mutants. So again, this is wild type, unstressed nucleus, upon stress goes to puncta. G68R, I already described, catalytically dead, still goes to puncta. NES, right? So you need to compare this with this, and you can see pretty much a large amount of the protein is in the cytoplasm. It goes to the granules, which would be expected because it's anyway supposed to be in the cytoplasm. The NLS, which is now predominantly in the nucleus, right? You don't see much cytoplasmic signal, you see mostly nuclear signal. When exposed to stress, oxidative stress, in this case sodium azide, likes to come back to the cytoplasm and accumulate in these puncta. The one which is tethered to the cortex is actually very poor in its expression. So under unstressed condition, it's actually very hard to even see the protein. But when you stress it, it forms these puncta at the cortex, right? So wherever it is, it tends to form these puncta. So this is the quantitation of number of cells forming granules and granules per cell. To cut a long story short, all the 3 mutants seems like they like to retain their ability to go to the puncta. So we like to think that this is something important for the protein, and no matter where you tie it to which part of the cell, it likes to go to the puncta. So forced change in localization of HMT1 does not abrogate localization of HMT1 to puncta. All right, so what have I shown you so far? HMT1 undergoes loss of nuclear localization, relocalizes to cytoplasm and accumulates in granules upon oxidative stress. It does not co-localize with P bodies, SBP1 or SCD6, and the catalytically dead mutant does not abrogate the localization of HMT1 to granules. All right, so this was pointing towards perhaps a non-canonical function of this protein. Now, what could that non-canonical function be, right? So Sakshi started looking at literature and tried to come up with different kinds of ideas, and after going back and forth with many different hypotheses and theories, she basically came across this publication that came out in 2017 from Susan Lindquist's lab, where what they actually did was they expressed synuclein in yeast cells and looked for modifiers of synuclein. And one of this modifier turns out to be HMT1. And what they found was that deletion of HMT1 enhances synuclein toxicity, right? So you get rid of HMT1, you express a human protein in yeast cells, and that modulates its toxicity. Now, on its face value, it sounds a bit uncomfortable because you're taking a human protein, putting it in yeast and asking, well, tell us what could affect the toxicity. So we were taken aback by this, but then we went back and looked at the literature, and there is a large volume of literature that uses yeast as a model system for not just synuclein, but variety of other proteins, including various neurodegenerative disorders such as Parkinson, Huntington, ALS, so on and so forth, where people have done screens, genome-wide screens, looking for modifiers of these proteins, and the hits that have been obtained from yeast have then been validated in other model systems such as C. elegans, mice, and even cells. So that made us feel a little comfortable that perhaps this is something worth exploring. So Sakshi decided to actually look at this seriously, that could this be true, and could this be related to the so-called non-canonical role of HMT1 that we are hypothesizing? All right, so alpha-synuclein, just a quick overview. It's a small 140 amino acid protein that has been implicated in Parkinson's. The Parkinson pathology is concerned with these Lewy bodies, which is because of these toxic oligomers. This happens when degradation of synuclein is misregulated, and that often happens in response to oxidative stress and mutations in some of these proteins which have been implicated in Parkinson's, right? So this is the sort of a small overview of alpha-synuclein. Before you ask me, alpha-synuclein does not have an arginine residue, right? So it cannot get arginine methylated, right? And of course, it causes this disease. So we started this project with the idea that we would find upstream regulators of translation repression, translation control, because we are looking at an enzyme that affects RNA binding proteins. But with these observations now, we are actually going in a very different territory, which we didn't intend to go to. But then I guess one of the excitement of doing research is following where your results take you to, which is what Sakshi did. Okay, so what do we see here? Oxidative stress causes localization of HMT1 to granules. Oxidative stress is also an important aspect of synuclein aggregation, right? So the obvious thing to ask is, could these two be related? Okay, so first things first, she wanted to confirm what they saw. Is it true in our hands or not? So this is wild type yeast strain with empty vector, grows the way it grows. You overexpress synuclein, overexpress in the sense it's a human protein, so you really don't know what is overexpression, underexpression, but yeah, it's expressed on a plasmid. So it causes toxicity, which is known, nothing new about that. You add delta HMT1 to that, and it alleviates the toxicity of, it enhances. No, wait. Yeah, sorry, no, it doesn't. So this is HMT1 deletion on its own behaves just like wild type, which is, there is no growth defect of HMT1 deletion on its own. And now when you combine HMT1 deletion and synuclein, you see that it grows worse than all the other three, confirming what was earlier reported. So this is good, right? Any of you relying on literature, the first thing to do is to confirm whether it works in your hands or not. Many a times it doesn't. All right. This is all significant. Now the question was that, can we put back HMT1 and rescue this toxicity, right? So again, wild type, we combine HMT1 deletion, there is a growth defect. You add wild type HMT1 back, which is this green bars, and it rescues. More importantly, you put back the G68R mutant, that also rescues. In fact, it rescues better than the wild type protein. All of these are significant, the green versus red and the purple versus the red, suggesting that the catalytically dead mutant, which still manages to come to the cytoplasm localized to puncta, is good enough to rescue this toxicity. This has been quantitated as growth rate, saying the same thing. This is the growth defect, which is rescued by wild type, and this is G68R. Okay, the question then is, does it affect protein levels, right? Because synuclein often, toxicity, as I suggested, is connected to its levels. This is synuclein levels in wild type cells. You look at it in absence of HMT1 deletion, you see there is a significant increase in protein levels, right? You put back wild type HMT1, they become more or less like wild type strain, where HMT1 is present. But when you put back HMT1 G68R, we pretty much do not see any synuclein signal. And when earlier Sakshi used to show this to me, I never believed it, but now we've repeated it several times. We still don't quite understand why there is no synuclein visible, but that's the result, and it kind of matches with the better growth curve that we see. But we still are trying to understand why this phenotype is the way it is. But this is a clear phenotype. So there's nothing there, right? All right. Levels are going down. Could it be affecting the degradation of synuclein, right? So she expresses Gal-inducible synuclein, which is how the original screen was done. Most of these screens are done by Gal-inducible plasmids, so we just followed that. And we did a chase experiment. And to cut a long story short, what she finds is that when there is no HMT1, there is decreased degradation of synuclein. So this is wild type, whereas this is HMT1 deletion, indicating that somehow HMT1 is able to affect degradation of synuclein. She did mass spectrometry analysis looking for partners of HMT1, and she found components of the proteasomal pathway come up in these different attempts that she looked at, suggesting that perhaps association of HMT1 with the proteasomal machinery could be important for this degradation. Buoyed by these results, she actually looked at ubiquitination of synuclein in absence of HMT1. And she found that when in the absence of HMT1, there is decreased ubiquitination of synuclein using this one antibody that we have characterized. We are still trying to get mass spec results for this, which we have so far not managed to get it. But this is some indication that ubiquitination goes down, which makes sense because the protein levels of synuclein seem to go up. All right, so what have I told you so far? The story is suggesting that under normal condition, HMT1 regulates synuclein protein levels, and that affects toxicity of synuclein, and that seems to be by somehow promoting the degradation of synuclein. We don't know how at this point. Since it affects toxicity, does it affect synuclein aggregates? So she looked at the aggregation of synuclein with and without HMT1, and she finds that this is how normally synuclein localizes. It's present largely at the cell wall because it's a lipid-binding protein, and there are certain specific lipids in the cell wall where this protein binds. This is not our result. Many people have looked at this and published this. But in delta HMT1, you not only see a general increase in the intensity, but you also see aggregates of synuclein, which is consistent with the idea that protein levels are going up, and thereby aggregation of synuclein seems to be going up. Does it happen in response to oxidative stress? Because remember, that was the key part, right? It comes to cytoplasm in response to oxidative stress. So she looks at H2O2 stress, recovery, and then live cell imaging. I'm not sure how much you are able to see here, but the idea is that in absence of stress, it is present, it aggregates more, which is what I just showed you. But in response to stress, also there is more aggregation visible in the absence of HMT1. We are now looking at synuclein. And even during recovery, although the synuclein aggregates recover to a large extent in wild type strain, but there is a lot more aggregates persisting in the absence of HMT1, which is what is shown here. So there is aggregation defect, but also looks like there is a disassembly defect because even during recovery conditions, we see more aggregates of synuclein in delta HMT1 as opposed to wild type. All right. Do these co-localize? If that's why HMT1 goes to these puncta, would they co-localize, right? So she looked at synuclein and HMT1 co-localization. it partially co-localizes, right? So we were quite excited about this result. We were like, all right, this is going to be the key experiment. We'll see a nice co-localization, and perhaps that's how it works, right? But that's not the case. We'd see some co-localization, but it's not complete. And I'll come back to that to try and explain that if I can. All right, since we found a disassembly defect, Sakshi then got interested in the idea that what if these are the sites where HMT1 causes disassembly of synuclein aggregates, right? So she tested the idea of doing the disassembly of synuclein aggregates with and without HMT1. So this is again a protocol that's standardized in the field where people purify granule-enriched fractions, incubated purified protein of interest, in this case HMT1, and they look at the partitioning of that protein in soluble and insoluble fraction. Partitioning in the insoluble fraction is equated with presence in the aggregates. So this is how incubation with the buffer looks like. When you incubate it with HMT1 wild type, or you incubate with purified HMT1 G68R, both of them actually reduce the enrichment of synuclein in these granules, right? So again here, for whatever it's worth, we find that G68R is significantly doing a better job than wild type HMT1 in terms of driving synuclein out of the granules, right? So we think that HMT1 perhaps can disassemble synuclein aggregates. Is it disassembly first and then degradation, or there is some other mechanism? So Sakshi did a time course experiment. So this is how it looks. So now we are looking at synuclein. This is how it normally looks like. You stress it, you can see aggregates, you let it recover, and what we find is that there is the aggregates disappear, and then slowly the protein levels start going down, right? So this is how it normally happens. When you look at this for the context of the granules, right? So percentage of cells forming granules, there are a maximum number of cells with granules upon stress, which slowly go down upon recovery. So by 10 minutes, there is already a decrease in the cells with granules, right? So which means granules have started disassembling. But when you follow the synuclein levels, it seems like they only show a significant reduction at this time point, which is the 30-minute time point. Both this and this are not significantly different from the stress that's this particular panel. So the reduced protein levels seem to kick in around 30 minutes post-recovery, whereas the granule disassembly seems to kick in at least in the initial stage by 10 minutes. So perhaps it's disassembly first, followed by degradation. All right, all this is in yeast, right? So of course, does this hold true where it matters most? So we then went on to the human homologue of HMT1, which is PRMT1. We asked the question, does PRMT1 change localization? Because that's where we began with, right? So PRMT1 under unstressed conditions is largely nuclear, as you can see here. In fact, the cytoplasmic signal that we see with yeast HMT1 is not very prominent with PRMT1. But when you stress these cells, right, either with sodium azide or with, and this is endogenous PRMT1, by the way, you can see that there is a lot of cytoplasmic relocalization of the protein, and it accumulates in these puncta, right? Again, nucleus to cytoplasm ratio changes, which is what this is saying. The granules assemble, and the PRMT1 levels actually go down, right? So this is not because of overall increase in PRMT1 levels. It's because, I don't know what it's because, but it's not at least due to increase in protein levels. Okay, synuclein localizes to puncta under unstressed conditions. This was known. She wanted, Sakshi confirmed this, that you express synuclein in these cells. They localize to these aggregates. But like in case of HMT1, could PRMT1 cause disassembly of synuclein aggregates? So now instead of yeast cells, we have the HeLa cells, which is expressing synuclein tag to M scarlet. We get the granule-enriched fraction, we add purified PRMT1, and we ask what happens. All right, so what we observe here is that when we add purified PRMT1, I apologize for the poor quality of the blot. We are still trying to get better versions, but we have repeated it 3 times. We see the same results, but I agree this is not the best blot that we could present here, but this is an ongoing story, as I told in the beginning. So when we add PRMT1 purified form, there is reduced amount of synuclein that's present in these aggregates. This is with the buffer. This is with purified PRMT1. In fact, PRMT1 itself localizes to these insoluble fractions, which is something we are trying to figure out right now. Why is that the case? But the important thing is that, like HMT1, PRMT1 can perhaps act as a disassembly factor for synuclein. All right, finally, synuclein causes toxicity, right? So could PRMT1 be able to rescue this toxicity? So what she did was she overexpressed synuclein in these cells, and you can see that there is a reduced viability of these cells. But when you express PRMT1 in trans in these cells, it rescues the viability of these cells. And again, very importantly, VLD, which is similar to the G68R mutant observed in case of HMT1, which is the catalytically dead mutant, rescues this toxicity, suggesting that, like in HMT1, this seems to be independent of its catalytic activity and perhaps a non-canonical function of this protein. This is the cell viability just by overexpression of PRMT1 and the VLD, suggesting that, if anything, there is a slight decrease. But of course, here we are seeing a rescue of the toxicity. There is no synuclein here. This is simply to ask the question that would these proteins on their own cause any kind of change in toxicity levels? All right, so with that, what I'm trying to suggest is that there are synuclein aggregates, and they usually cause cell death. What our results, which we didn't seek out to find in the beginning, but they have taken in this direction, is that this methyltransferase, independent of its catalytic activity, is involved in the disassembly of these synuclein aggregates. And post-disassembly, they are also promoting ubiquitin-mediated degradation of this synuclein protein. And by doing that, they are able to rescue the toxicity, which is what we are observing both in case of yeast as well as in case of humans. So with that, I'll stop here. IISc has been a fantastic place to do science. Eternally thankful to this group, which is a very hardworking group and allows me to present these results. Funding agencies have been very generous. And again, Sakshi has spearheaded this work. Thanks to all of you, and I'll be happy to take questions.
Did you look at the PRMT1 puncta and did mass spec there?
In mass spec? No. So we are managing to purify or enrich these puncta. We are in the process of sending these samples.
My thought would be that there may be many other proteins that it may be.
Absolutely, yeah. So I think the point being raised is that, is this phenomenon just for synuclein, right? Very likely not, right? So what I'm suggesting here is that HMT1 or PRMT1 has a chaperone-like function, perhaps. And that may not be just limited to synuclein, but perhaps other proteins. What are those other substrates? What is it doing in yeast, right? I haven't managed to tell you about that. So that is where this question goes to. We are trying to look at that. Yes. No, I mean, so yeah, so if I may do a hand-waving argument here, the point is that because G68R or VLD seems to do a better job, perhaps there is a competition between these two jobs that HMT1 does inside the cell, which is to methylate proteins with RGG motifs and this non-canonical function. What G68R does is that it frees it of that other job so that it can focus more on this non-canonical activity, and that's why it seems to do better in the assays that we have looked at. Of course, we need more evidence in that direction, but intuitively that makes sense, right? Because these are two competing things. When you are in the cytoplasm, you can't be in the nucleus, and at least by microscopy, it doesn't look like it's in the nucleus and vice versa. So perhaps that's the explanation, but I don't know entirely.
Sir.
So can you comment that why the catalytic dead mutant was rescuing better compared to the the wild type mutant? And is it known that HMT1 binds to ubiquitin or acts like independently of its catalytic activity, acts as a E3 ligase or something like that?
Yeah, the first question what was what I just responded, that perhaps there is a competition between the canonical and non-canonical function. And therefore, when you free of its main day job, then it does the night job better, if it were to be that way. The second part, I don't even know if it acts as a ligase right now. Right? All we see is that there is reduced ubiquitination. It could be direct, it could be indirect. Right? Perhaps like Umesh is pointing out, there are things in that puncta, right, like ligases, which are being recruited by HMT1 or PRMT1 onto synuclein and other kinds of proteins by simply acting as a scaffold or some such kind of activity. I don't know the answer to that. It does not have an arginine. So we are safe there.
So under no stress condition, HMT1 methylates RGG domains in several RNA binding proteins, right? How many substrates and what does that do to these RNA binding proteins under no stress?
Yeah, so the proteins that we have looked at are mostly cytoplasmic proteins which act as translation repressors. These are basically 4G targeting proteins that repress translation by binding 4G, what Tanweer was referring to. In one of the cases, we find that a specific stress increases the methylation of that protein. For another protein, it actually reduces methylation. So I am unable to give you a theme-based answer that methylation promotes interaction or reduces interaction. I think that's based on the context of that mRNP.
But the point is that when you get rid of HMT1 or you express its catalytic dead, you're likely to affect the activity of many different RNA binding proteins, including some in the nucleus, which have RGG domain, and that includes nucleolar proteins, ribosome biogenesis, everything.
Yes, yes. But like I said, this is something that's what we are able to tie it to is its localization to those structures where none of these proteins seem to be present, right? So what it's doing in those structures seems to be independent of its substrates. So yes.
Yeah, but there's no evidence that the puncta of HMT that you see are functional in any way, right? I mean, that's just an effect
they are
of
dynamic.
I mean, right, when you do oxidative damage, it's probably killing the enzyme. And so now it's aggregating in some way, right?
No, so that's what I showed you in the first slide, that it is dynamic. You take away the stress and it reverts back to its original localization. So it doesn't seem to be an endpoint that's dead. And these stresses are for only 30 minutes.
Did you actually show that the protein that was in the aggregates comes back out? Or is that new protein that's made out?
30 minutes of recovery, during which, in response to stress, there is global translation repression. There is a formal possibility that newly synthesized protein could now be going to the nucleus, but these stresses that we are using are used by many people in the field and they do not cause cell death. These are transient stresses, which allows the cell to return back to normal metabolism. So that's my argument at this point. Okay.
Yeah, no further questions. Let us thank Raj.